All right, class, we're taking notes now on the bottom of page 109. The figure is early morphogenesis in an animal. And in this case, we're talking about any animal at all. So we're talking about generalizations. In invertebrates, the only difference would be that cell membranes wouldn't be forming, so these drawings would look different, but the same phases would apply. So, as soon as fertilization has happened, then we have a zygote, a fertilized egg. All right. And the zygote has an animal pole at the top. This is what developmental biologists have called it, and a vegetal pole underneath. Now, depending on which sort of animal it is, okay, post-fertilization or potentially even before fertilization, something called cytoplasmic determinants, the timing of cytoplasmic determinants depends on the animal. Determinants. Effects. Okay. So cytoplasmic determinants are chemicals found either, as early as in the fertilized egg, which is the zygote at the beginning, in the case of some frogs, or as late as the 16-cell stage. Cytoplasmic determinants are chemicals that are unequally distributed, and they control gene expression through gene packaging. Okay, so the timing of cytoplasmic determinants varies based on the animal. All right, so this fertilized egg, or this zygote, is an embryonic stem cell, which we've talked about before. It has the potential to be every single body part of an organism fully formed. Now, at some point, depending on the organism, one of the sides of this zygote becomes pigmented. The cortex around the cell becomes pigmented differently than the other side. And this is why the top is called the animal pole and the bottom is called the vegetal pole, is early developmental biologists noticed this pigmentation. So a series of cell divisions happen that we call cleavages, okay? And these are mitotic divisions. So you're going to go from an embryonic stem cell to 2 cells, okay? And in some organisms, there might be a 3rd layer of pigmentation already, but in all organisms, eventually there will be. You'll go 2 cells, you'll go 4 cells, you'll go 8 cells, okay? And this second pigmentation, and what I'm trying to draw here, or this third, pigmentation is called the gray crescent. So this is my 8-cell embryo. Now, during these cleavages or mitotic divisions, okay, the embryo isn't changing size. It's just increasing cell number, which is kind of interesting, I think. All right. So we're going to move from, these are whole mounts, meaning we're looking at the whole organism. And this will become relevant when we start looking at more slides. We're going to move from that kind of drawing to a cross section. So at some point, this organism is a ball of cells, anywhere between, 16 and hundreds of cells. Okay, after the organism goes through the morula phase, a cavity opens up in the center. This cross section again. and the cells move to the outside edges. And then it's called a blastula. The blastula can be recognized in our slides because it has this opening called the blastocoel. Okay, so this is the blastula. All right, now, cell divisions continue, mitoses continue, until finally you get to the stage called the gastrula. The gastrula is pretty much one of the coolest parts of development because this is when things really start to happen. So at the point of a gastrula, we're talking about thousands of cells. So I'm not going to draw cells anymore. Instead, because of what happens during gastrulation, I'm going to draw these embryonic tissues. So during gastrulation, the embryo is still the same size. It's going to form 3 transient embryonic tissues. Oops, one little yolk plug right there. All right. And most of the tissues we're going to be talking about are transient for a while until, right, we start to form adult tissues. It forms these 3 transient tissues called the 3 germ layers. So at this stage of development, the blastocoel's gone. It's been replaced by a new cavity called the archenteron. All right. In addition to that, this cool thing that we're going to talk about on page 110 happens, where we form these 3 germ layers. So on the outside here, we've got the ectoderm. And in a weird way, it's sort of, I mean, I know this is a circular embryo, but it's sort of like the outside tube of the organism. Right, really, if we squash this and elongate it, it's really just a tube. So in my drawing, the ectoderm is going to be blue here. All right, that's the outside layer. The middle layer, so ecto means outside, it's kind of nice, or out. All right, the middle layer is called the mesoderm. Meso means middle. So in our drawing, that's this black layer. That's the middle. All right, and the endoderm. That's the inside. It's the inside tube. And I've just left that not colored. All right, so gastrulation happens, and a lot of chemicals are affecting gene expression and giving cells partial identity. We've talked about this. We're going to keep talking about it, right? By changes in gene packaging, even though every cell, so this is gastrulation, is the process during which the gastrula forms. Right? This is really changes in gene packaging that are then inherited and cells gain identity. It's pretty awesome, actually. All right, so finally, the organism starts to grow during organogenesis and organs start to form. So pat your head. At this point, you have an anterior, say anterior side. Pat your bottom, you have a posterior side. Rub your belly and say ventral, you have a ventral side. Pat your back, and you have a dorsal side or a back side. You also, of course, organogenesis means building of organs. You're building organs. A lot of master regulatory genes like Hox genes that we've talked about are active during this phase and start to construct not only internal organs like muscle and bone, but also limbs and extremities. You also have a right and a left at this point. All right, so next we will talk about cortical rotation, which is, and the initiation of gastrulation on page 110. All right, class, so now let's talk about gastrulation in more detail, and cortical rotation, which is the animal cells moving down to cover the vegetal cells during development, or at least that's the last part of that. So I suggest the way that you do this section is you first take these notes, okay? And this is on page 110. And on the left side, you'll see these diagrams, okay? The cells are moving and these drawings don't move, okay? So it's kind of difficult to explain without physically being there to like gesture and everything and tell you how it's going or what's happening anyways. So what we're going to do here is you're going to take these notes and then you're going to use this video to help you understand what's actually happening. All right, so gastrulation begins when this small indentation forms right here called the dorsal lip of the blastopore, the DLB, and it forms in what we're going to talk about, we're going to call the gray crescent later. We're also going to refer to this as the Spiemon organizing center, this general area of the embryo. It's really important because it gives instructions to cells. So the dorsal lip of the blastopore forms under the leading edge of the animal pole, okay? And these animal pole cells are moving down the outside of the embryo over it, over the vegetal cells, okay? And at the dorsal lip of the blastopore, okay, cells are pushing in interiorly, all right? So as the dorsal lip moves vegetally on the embryo, cells are moving in here, okay? The cells that turn first, right, closer to the ectoderm up here, they're going to form mesoderm. The cells that turn further in, they're going to form endoderm, and they're also going to make this new cavity called the archenteron, and that's going to replace or push out the old cavity, the blastocoel, which is the identifying feature of the blastula, which is what this thing was at the beginning of gastrulation. Okay, so like I said, these cells are moving down, which means the dorsal lip is moving down posteriorly. All right, so at the end of gastrulation, okay, the dorsal lip has moved all the way posteriorly, and sticking out of it is this endoderm-lined cavity, the archenteron, and endoderm itself at a position called the yolk plug. All right. After gastrulation, organogenesis begins.